Morning, everybody. Hopefully you're all having a beautiful sunshine out your windows this morning. My name's Narman Kassam. I'm the chair and zone clinical department head for the Department of Medicine. I would like to start by respectfully acknowledging that the University of Alberta is located on Treaty 6 territory, traditional lands, and gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to enrich our vibrant community. The Dean's Lecture Series, Precision Health, takes place several times throughout the year and invites experts and thought leaders on various topics to deliver a lecture in their field of expertise. Thank you for joining us today as the Department of Medicine hosts this iteration of the Dean's Lecture. And I would now like to introduce our Dean, Dr. Brenda Hamelgarn, who will introduce our speaker today. Thanks.
Thank you very much, Dr. Kassam, and thanks to the Department of Medicine for sponsoring this event. I have the great pleasure of introducing our guest speaker, Dr. Hayden. Dr. Michael Hayden is a Killam Professor at the University of British Columbia and holds a Canada Research Chair in Human Genetics and Molecular Medicine. He also received an honorary degree in science from the University of Alberta in 2009 and thus has close connections to the university. He's the author of over 900 peer-reviewed publications and invited submissions. And Dr. Hayden has focused his research primarily on translational medicine, including genetics, lipoprotein disorders, Huntington's disease, precision medicine, and drug development. Dr. Hayden and his research group have identified 10 disease-causing genes, which includes identification of the major gene underlying high-density lipoprotein in humans. Dr. Hayden has been the most cited author in the world on ABCA1 and Huntington's disease since 2006. Dr. Hayden is the co-founder of 5 biotechnology companies, and he currently sits on a number of different public and private boards of biotechnology companies. And he was named, in fact, one of the 50 Canadians born in the 20th century who've changed the world. Dr. Hayden is the recipient of numerous prestigious honors and awards. Most recently, he was awarded the David Dubinsky Humanitarian Award from the American Friends of Soroka Medical Center. He was inducted into the Canadian Medical Hall of Fame in 2017 and was named one of Pharma Voice's 100 of the most inspiring people. He's also been awarded the Order of Canada and the Order of British Columbia, to name but a few. Dr. Hayden's also committed to empowering others, and in addition to mentoring over 100 graduate students and postdocs, he's also a TED mentor. So with great honor, I'd like to introduce and present to you Dr. Michael Hayden.
Well, thank you. Just in terms of bandwidth here, I'm going to stop my video, but I did want to say how thrilled I am to again be visiting in one form the University of Alberta. I want to thank Dr. Hamelgarn and Dr. Kassam for giving me this opportunity, and also Dr. Sikorsky for inviting me. And I will just stop the video in an effort to make sure that you can continue to hear me. So I'm delighted to give this talk today because it really talks about my own experience. And when we talk about precision medicine, often the precision medicine has been really understanding patient presentation, the cause for the disease, and how this has led then to approaches to drug development and translational medicine. So I want to share with you a few personal perspectives and take you on a journey. So the first thing is that many times as a physician scientist, you're facing the word no. No about how difficult it is. No from family who's saying you've got to give up some things. No from the difficulty of taking an academic finding and thinking about ways to translate this to something and products and services for patients. So just to share with you where my own journey began, I was born in South Africa and grew up and went to medical school and physician scientist there. Very early on, in my career, I learned that you don't have to be at Harvard and Stanford to make discoveries that have impact on the world. This was Chris Barnard, who came to, and this is his son, who was in my medical school class, who illustrated for us how even places that are geographically distant can have very significant impact. And he did the first heart transplant in the world. I was moved. I was brought up by a single mother and very much taken to talks. And this was the talk from Robert Kennedy when he came to South Africa against the government in 1966. I was a schoolboy. My mother told me, come out of school because you learn much more from this talk, which was at the University of Cape Town. And at this talk, Robert Kennedy, and this is actually on his grave, this is what he said. And he said, each time you stand up for an ideal or act to improve the lot of others, we send forth a tiny ripple of hope, and crossing each other from different centers of energy, these ripples bend a current that can sweep down the mightiest wall of oppression. And certainly in my life, oppression has been really, of course, living in South Africa, that was one example, but really oppression is all about illness. Illness stops you from being fully who you are. Illness keeps you captive. Illness stops you from enjoying daily activities of life. And so I've really kept this ripples of hope very alive. Of course, during South Africa, and this was in the mid-70s, we were very involved, and we really had to take on lots of particular challenges. So I went from the University of Cape Town, where I did my initial training. Then I went to Harvard Medical School, finished my internal medicine at the Brigham and Women's, and then eventually came to UBC and founded the Center for Molecular Medicine, which was really committed to discovering and translating precision diagnoses, which is diagnosis by DNA assessment or some molecular assessment, and trying to translate that into therapeutic approaches. Of course, this is happening at a time where genetics is everywhere. These were some of the this was in the early 80s. The first draft of the human genome was actually in Vancouver in 2002 at a human genome meeting. And so this influences a lot of the development. Now, one of the key things for me in thinking about drug development, how we learn, is really thinking not just about the regular, but the irregular, the black swan event in biology. And the black swan event in biology is important because that black swan is a patient who is unusual, a patient that often has a molecular diagnosis. They often outliers. These mutations have extreme impact and in a way are predictable as we learn more about this. So these black swans become targets that are clearly not compensated for in humans and allow you to have novel, validated targets for drug development. Now, when we think about drug development, it's still difficult because in phase 2, the rates of attrition are still significant, 66%, mostly due to inadequate efficacy. And in phase 3, the biggest issue again is insufficient efficacy. So efficacy is still the major focus, and it's the major barrier towards drug development. And the key is finding ways to overcome that. And this is where precision medicine and genetics plays a role. So it's very important to go fishing where there are fish. And where there are fish is seeing the unusual patient, the patient who doesn't respond as normal, the patient who's not, and these occur in families. So often, for example, there are patients who are resistant to pain or resistant to obesity. And most gene mutations cause loss of gene function. And if you can identify the gene, a gene blocker can represent that desirable phenotype. And I'm going to give you a few examples today around what we've done over the years. Now, some of the examples actually came from Canada. And this was the late Francis Plummer, a brilliant and amazing scientist out of Winnipeg, who initially identified resistance to HIV infection amongst patients who were clearly and subjects who were clearly exposed. And these particular people had changes in a particular receptor, CCR5, and this led to the development, and this CCR5 was lost and not functional. This led to the treatment of CCR5, early development of treatment for HIV and receptor antagonists, because this protein blocks the entry of cells blocks the entry of the virus into the cells. So this was one example of black swans. Now in South Africa, and this was as a medical student intern, this was my mentor and professor at those times, there was a whole group of patients with unusual phenotypes. Here was patients with very tall, and they also had increased bone density. And I saw some of these particular patients. So this is all from the same family. This is called sclerosteosis. But was interesting, and these patients, when you looked at the X-ray, they had increased bone density. And this made you think that these patients, some loss of function or some mutation that resulted in increased bone density. And when you looked at the X-rays, this doesn't look like osteoclast abnormalities because it would be patchy, but rather looks like something regulating the osteoblast, because the osteoblasts look overactive. And as a result of that, I was able to get DNA from these families and be able to help with a group in Seattle to try and identify that particular gene. The gene itself was shown to be a loss of function of a key protein, and the key regulator for the osteoblast in humans. And what happened, it's a negative, it was a repressor. So if you, in fact, inhibit this, you can increase bone density. And this resulted in the development of monoclonal sclerostin, because it comes from sclerosteosis antibodies. And these were patients in clinical trial for treatment of osteoporosis. And this was a project that was essentially licensed to Celtech in the UK, which became UCB, and also to Amgen in the US. And what happened when patients were treated with this humanized monoclonal antibody, they actually had increased bone density. And eventually, this was published just a few years ago about the treatment of this for low mineral density, and resulted in a potential, as they said in the editorial, potential breakthrough in osteoporosis. And this was actually then approved and now is on the market for treatment of osteoporosis in the United States, Canada too, I understand, as well as in Europe. So this is an example of a rare genetic trait that gave insights into a pathway that could be only illuminated by understanding the mutation that occurred in these families. Now, in addition, there are many other examples where one looks for the rare exception. And let me share with you what we did in pain. So, of course, pain is a big problem. The number one complaint bringing patients to doctor's office still, and of course, this has led sadly to the terrible addiction and overdose with opioids. And so we were very interested to study patients, and this is from the New York Times, just looking at patients who actually felt no pain. And we knew about those patients in Canada because there were stories of families, particularly from Newfoundland, in isolated communities where there was a lot of consanguinity. These were patients who said childbirth was not painful, they had numerous fractures and had no pain. And generally, these were patients with essentially congenital insensitivity to pain. So we identified families. This was the initial family from Newfoundland. We identified families, again, for this exceptional opportunity. And we were able to find that these patients all had loss of function mutations in NAV 1.7. Now, we were helped because a lot of these patients had truncation mutations, so they had no protein. And we knew that this became then the primary drug target for development of novel approaches to pain. I would say there are numerous, this still remains the number one target, both for humanized monoclonal antibodies as well as oral therapy for treatment of pain. Further validation of this came from, so this is loss of function, came from patients who had a gain of function. There's another group of patients who have gain of function, and these patients have excessive pain. So this would also be very helpful in these patients. Targeted precision therapy, this is called erythromelalgia, not a common disorder, but patients have significant pain. They end up really with such pain that suicide is quite common. And this remains a very significant issue for these patients, where an antibody or an antagonist to NaV 1.7 would be life-saving. So just to share with you, these are some of the families. These were spoken about in the mid-2000s, the first decade in the Globe and Mail. These families, they told the story of anguish, suffering, and discovery of this particular gene. And in addition, around the same time, I started seeing patients at Children's Hospital who had abdominal pain. And this was serious. They had abdominal pain. And these patients presented at Children's with abdominal pain. And when we looked at their blood, they had extreme hyperglycemia, hypertriglyceridemia, or chylomicronemia. When we looked further, we actually were able, in this stage, actually even by Southern blot analysis, here you can see the normal gene, and here you can see patients who have two mutations. Well, a mutation here, this is a deletion, here's an insertion. And we were able to describe this, and this became the molecular basis for type 1 hyperlipidemia caused by mutations in the LPL gene. And we immediately, and essentially because these patients had presented, some of them in pregnancy that precipitated the outcome, really started working on mutation, on therapies for these patients. And lipoprotein lipase, this is an important enzyme. It normally breaks down triglycerides. These patients had massive increase. You can see they get xanthometer. They also have abdominal pain and often presents in children with failure to thrive. But we had seen a lot of patients with pancreatitis, and they can be, and this can be fatal as a result of the complications of pancreatitis. At the same time, we had heard that LPL deficiency was actually quite common in Quebec. And we started working with our colleagues in Quebec, where the frequency was very similar to that seen in cystic fibrosis, the highest frequency in the world in the eastern Quebec, around 1 in 40 carriers or 1 in 4,000 affected individuals. So we started trying to develop therapy. Lipid-lowering drugs were not effective. Enzyme replacement was not effective because the LPL half-life was too short. We wanted restriction of dietary fats, but this was really difficult. And then we started thinking, well, if we could replace this gene and get it expressed in the right area, we may have an approach to treatment. Now, the question became, what LPL formation would you use? At the same time, we had discovered that there was a gain of function in the LPL gene as carboxy truncation of 2 amino acids. And this truncation was associated with gain of function, what we called super LPL, increased HDL, protection against coronary disease, improved blood pressure. And so we actually recognized if we could use this gain of function as the formulation for LPL, that this would be potentially even more helpful, but you could go with lower dose. So this super LPL was associated with decreased triglycerides, increased HDL, and reduced risk for coronary disease, and this looked good. So here's the LPL actual formulation. You can see it's a truncation mutation. This protein has 449 amino acids, and this actually, it's a truncation mutation after these lysines, and so this is really the mutation we use. Now, we knew it was more effective because when you took animals, mice, that were gene targeted to have no LPL, these animals died in the first few days of life as a result of chylomicronemia. Furthermore, when you used LPL wild type, they lived a little longer, but only till about 22 days. However, when you use the same adeno LPL, but now use this formulation, this was really, you got almost complete rescue. So this told us, even in vivo, that this mutation, this gain of function that was detected as a result of mutation analysis in patients was really most effective. So by 6 weeks of age, these animals really normally would die. And so we began AAV gene therapy using LPL. And here's the construct. It has the truncation mutation, and we used AAV1 as the vector associated with it. We were able to show in mice quite beautifully in the early 2000s that you got complete clearing of plasma. These were patients with lower dose, and this is the wild type control. This was featured on the cover of Human Gene Therapy. And so this became the approach that we took. So here you can see in animals, for 1 year, single dose, treated these animals, and they had complete clearing compared to control. This was very encouraging, single dose associated with this. At the same time, we discovered a naturally occurring model that came from New Zealand that was a large second species with LPL deficiency. You can look at these cats' eyes. They have chylomicronemia. You can look in the fundus. And this was their plasma. And they had, again, a novel mutation. They had a complete absence of LPL protein and LPL activity. And so we also did gene therapy in these animals. By the way, many of them died from pancreatitis. And again, you can see here, treated with these, they had to be treated with the immunosuppressives in an effort because this was the human gene in animals that had never been exposed to LPL. But you can see we again got complete clearing. So we started phase 1 gene therapy for LPL, initially in the Netherlands, but we also had discovered and identified together with our colleagues, Dr. Gagne and others in Chicoutimi, large numbers of patients. And so the phase 2/3 study was initiated in Canada with Dr. Daniel Godet and also other colleagues in Quebec. This was the lead clinician in this study. Now, what we found with this study is that numerous patients did very well. They had significant reduction in triglycerides, and the incidence of pancreatitis was reduced dramatically. And there was no antibodies, there was reductions in xanthometer, and patients did extremely well. This led to the first approval, actually, and we didn't realize this at this time. This was the first approval for any human gene therapy in the world, and this happened in 2012. Approved by EMA, indicated for patients with familial LPL deficiency. You had to do this by precision medicine, genetic testing, and this was indicated for patients, specifically patients with no levels of protein. And so this was the first gene therapy. This was heralded, thought to be a blockbuster, but there were numerous issues associated with this. These were patients, this was featured in Canada. Here's a woman who went public with this. She said about pancreatitis, like acid going through, and then post-treatment, she was part of the trial, and 10 years later, turning point in my life, she had 2 miracles, and she had suffered from debilitating pancreatitis, hospitalized more than 40 times, and then post-treatment, 10 years later, has never had another pancreatitis attack. However, this drug was only kept on the market for a few years. And in 2017, at the time, it was the most expensive drug. This was marketed and licensed to Unicure that charged €1 million for this. And basically, it was hard to get insurance at that time, and the usage was limited. And so the company withdrew the project. But let me so this was sad, and particularly sad for people in Canada who had contributed to knowledge, and this therapy was not available for people in Eastern Quebec. And so the National Research Council of Canada actually came, and they said this was the world's most expensive drug, but now the drug is getting a second chance. And Brenda Potter here said, we've been fighting for 10 years, and now this is a new project ongoing in my lab together with the National Research Council of Canada for an improved version of Glybera, a different approach. It's going to be manufactured in Canada, and we are going to offer this at cost to all patients with LPL deficiency in Canada to make this affordable and accessible for patients in this country. So we're ongoing. The studies are going very well. We've got a formulation using a different vector that is superior to what was used before, much more potent. You can go with lower dose, and we'll keep you informed about all of these. So finally, let me share with you a little bit about a disease that I first saw in South Africa as a medical student, and this is Huntington disease. And in a sense, this was my opportunity to visit patients. This is caused by a CEG repeat in exon 1 of the gene. This is described by William Osler as the worst disease known. And it's the worst disease because it strikes you in the prime of life, and it's had inexorably progressive to death, usually 15 to 25 years from onset. In the US, there are a lot of patients with this disease, even though it is a rare disease and it's an orphan disease. 100,000 patients, 40 in US, about 4,000 in Canada, maybe, and in Europe also significant numbers, 40,000. Rest of the world, maybe 20 to 30. For every affected person, there are 3 people who are destined to be ill but not yet ill. And there is only limited treatment. When I was working at Teva, I took the deuterated tetrabenazine and led that development called Austedo, which is available, but that only treats chorea. And what patients are really asking for is something that can have impact on the inexorable decline associated with this disease. So in South Africa, I ended up, because it was said, again, everybody said no. They said Huntington disease does not exist in Africa. So as part of my PhD, I went to look at every hospital, mental health hospital, and was able to go through records and get appropriate consent. And we were able to find that the disease in persons of Caucasian as well as mixed descent was very similar to the frequency seen around the world. Huntington disease was very much alive and well, and sadly affecting families in South Africa. And these were some of the families I met. These were families disenfranchised by apartheid, but also disenfranchised by this disease. And I have permission to show these, as these were a source of great inspiration from these families. So here's AB, and this is his family, and visiting them living in poverty, but really welcoming me into their home. Now you can see it's a long time ago because that's me. And this is the family. He was a postman and welcoming me. And I was moved by the dignity, the determination, the resilience, and the honor that these families manifest, and their generosity of spirit. As part of this, I also got to know Marjorie Guthrie. She was the founder of the Huntington Disease Association of America. And here she is with Woody Guthrie. This land is your land, with Arlo here, a famous singer, most famous for Alice's Restaurant. And Marjorie encouraged me to come to the United States and actually helped me get through the immigration. And it was really because of Huntington disease and Marjorie that I eventually left South Africa. And here I am with Marjorie. Marjorie became a huge supporter for me to continue my work. So as you know, for Huntington disease, it's a autosomal dominant. So you have one normal gene and one abnormal gene. And this is caused by polyglutamine expansion and gain of function toxicity. And so what you can see, there are different approaches that have been used now for treatment. There's what's called knockdown of the mutation itself, the protein. This is where you can use non-selective silencing or silencing that targets just the mutant allele. So now you get selective or allele-specific silencing. And certainly in my lab, we've worked a lot on this for the last over a decade. And what you can see, the mutation can be silenced by targeting SNPs, single nucleotide polymorphisms that reside in the gene. And if you look at SNPs, they're on either side, and it targets the messenger RNA to cause degradation of that protein. So when you do that, you can actually knock these down. So when you use selective silencing or not, it's the same thing. For selective, you use an ASO that binds the message and results in degradation. Or if you use non-selective, it has impact both on wild type and mutant. And this was the approach that led to a trial by Roche as well as Wave recently. Some of this work has continued at the University of Alberta with Dr. Sikorsky in these trials. But sadly, there has been some recent news that I'll just share with you. So these were animal studies where we also showed here that you can get selective knockdown of mutant huntingtin while preserving wild type. Here's normal wild type huntingtin. You can see you can knock down just the mutant. And these gave encouragement. And also there's improvement of phenotype. So this gave encouragement for these studies. This is given into the spine, intrathecal. Of course, there were challenges associated with intrathecal, both the procedure itself, but also Huntington disease is caused by deep, initially early selective neuronal loss in the deeper parts of the brain. In later stage, it becomes a diffuse disease. And so here, this was the findings shown by Roche recently, and these were very unfortunate and devastating for the community. This is an endpoint for looking at progression. And here you can see patients both on treatment at Q8 in blue, every 16 weeks or placebo. And here you can see that the patients treated, particularly at Q8, are doing worse than placebos. So this is the ultimate result that one never wanted. Premium non-narcoa, firstly, do no harm. Sadly, these patients were doing worse. And this trial has now been stopped. And the allele-specific knockdown trial with Wave has also been stopped because they did not get target engagement even at high dose. So the reasons for the failure, I would say it's not completely sorted out. Firstly, this could be the fact that you also got knockdown of wild-type huntingtin. And wild-type huntingtin is neuroprotective. So you want to be able to also, this may be making you more susceptible to the gain of function from mutant huntingtin. You may not have got target engagement in the deeper layers of the brain. We also know that the ASO induced a neuroinflammatory response. There was CSF pleocytosis with white cells in the CSF associated with decreased CSF flow and ventricular dilatation. So that was a problem. And you also, there's an exon 1 that is an alternate splice that also was knocked down. So we also started working on other approaches to try and treat Huntington disease. And what you can see for all these devastating neurological and neurodegenerative disorders, they all have similar mechanisms for neuronal death. Spine impairments, alterations in calcium, BDNF deficiency, mitochondrial dysfunction, neuronal injury, and decreased autophagy. Now, we've got a drug that worked on the sigma-1 receptor that appears to positively influence these pathways. We know its target is the sigma-1. This was initially thought to be a dopamine modulator, but this is in vivo human targeting with fluspidine, which is a sigma-1 binder in the brain. You give fluspidine, you can see very nice lighting up of the whole brain. Then you pre-treat with your drug, and you see how much is obstructed, and here you see almost completely. That tells you that this drug is binding target engagement, firstly throughout the brain, and secondly at the sigma-1 receptor. We knew already from other literature that activation of sigma-1 is neuroprotective through all the mechanisms that are dysfunctional in Huntington's and other neurological disorders. And we further have shown that pridopamine can have impact on each of these pathways. So looking at this, there was a phase 2 study done whilst I was still at Teva, and we were and we extended the study once we knew the study was looking potential on impact on progression to go to 52 weeks because TFC is only measured in annual decrements. And this particular is now accepted by the regulators as a single acceptable primary endpoint. And it measures those things that matter to patients. How long can you work? Can you manage your finances? Are you doing things at home? can you dress and feed yourself, and who takes care of you? And this is accepted as primary endpoint by the FDA. So this was the data that we showed in our analysis, post hoc analysis, looking at early HD patients, that the placebo went down as expected. But I would say, surprisingly, Huntington patients did not show any progression, or certainly less than the placebo. And it was this that led both the European and the US regulators, and now also Health Canada has allowed this, and this is now a phase 3, single phase 3 placebo-controlled study. It's ongoing. The target is early HD. The primary endpoint is TFC, and the first patient was randomized in October 2020. Now, just to say, also, this study is actually ongoing. We also were looking to do this in Alberta. Edmonton was at already maximal capacity with Roche and Wade. So this study is actually being conducted in Calgary, as well as other sites throughout Canada as well. Now, at the same time, there was a recognition that the sigma-1 may also be associated with ALS. So again, precision medicine, because there were patients that were rare exceptions, black swans, who had mutations in sigma-1 that developed ALS. Some of these patients had complete loss of function. This is a frame shift mutation. And this caused a juvenile form of ALS. So the worst, if you are absent sigma-1, you get ALS. And if you have, however, a missense mutation, resulting in a dysfunctional protein that's just partial, you get an adult onset ALS. So this gave us significant hope that also these drugs may be useful in ALS. Now, we looked at SOD1 mice. These are mice, and this is done through a small company that we founded called Prolenia. And here you can see this is what happens in ALS. You get immobile myofibers. But you want to convert these into contracting myofibers, and this is an in vivo preclinical model for efficacy. Here you can see in wild type, about 70% of myofibers are contracting. If you give predopidine, you see you can get even higher, up to 80%. And in SOD1 mice, you get a loss of function mutation here, and these animals have only 50%. Half the myofibers are immobile, and half of them are contracting. You give the drug itself, and what happens is you can convert immobile myofibers to contracting myofibers. And then if you ask yourself the question, is this mediated by the sigma-1 receptor? So you cross these animals to sigma-1 and do the same experiment, and what you can see is that you get no effect. So the effect is completely deleted if you remove the sigma-1. That tells you that this is operating through sigma-1. So what you can see, this study is now part of, again, a phase 2-3 study in ALS. This is being led by Merit Kudkovitz out of Mass General. And actually, Mass General got a donation to foster innovative drug development in ALS. And they've chosen to include this as part of their study. So we now know that predopidine can have impact on all of these, improving autophagy. All of these are now published, and that's allowed us to do this. So I would say, as I reflect back on my own career, it hasn't been following the tracks necessary of those that were laid. It's very much laying your own tracks. There are many obstacles, there are many opportunities, but as we focus also on translation, often the involvement with biotech and pharma is crucial to do the rigorous implementation of clinical trials that would allow approaches to really get these to patients. So I think the whole story that I've spoken about is really about from genes to therapy, from understanding the molecular basis in those patients and translating that either in drug development or in gene therapy in an effort to focus on bringing novel approaches to therapy. And important to note, a lot of these findings were because you were able to define the molecular basis, define the patient very well from genotype to phenotype, and then translate that to therapy. So this has been a great journey for me. And I very much appreciate the opportunity to talk with you today and to share some of these insights. I know I've spoken about a lot of opportunities, but very much appreciate the opportunity to share some of this with you this morning, and particularly as you are my alma mater too, having been awarded a degree from the University of Alberta some years ago. So thank you all very much for your attention.
Thank you very much, Dr. Hayden, for that excellent presentation. You really have shown how, it is from genes to therapy, how a rare genetic trait can actually be translated more broadly into benefits that can be applied to a more broad population. So thank you for that. We will be open to questions, so if individuals could please type their questions into the question and answer box. I'm just going to start off with one question, though, for you, Dr. Hayden, and this is something that we hear a lot about, and that relates to gene therapy being one of the most expensive types of therapy that there is. And you spoke very briefly about Glybera being reinvented and how you're now able to offer it at cost, affordable, and accessible. And I wonder if you could just speak to that a bit more and how you're able to do that. I think there's many lessons learned there that many of us could benefit from.
Yeah, well, thank you. That's such an important question. So in the old days being 2012, so it's not that old, but it's old for particularly around manufacturing. Manufacturing has come down in cost logarithmically. So for example, if you're going to grow up this vector in a small 20 or 50 liters, this will cost you just to manufacture it around $200,000. So all of a sudden, but if you go to 1,000 liter or 2,000 liters, you're able to reduce the cost per dose to $15,000 or $10,000. So dramatic. So the key is manufacturing expertise to reduce the cost and also not taking profit. So that becomes, and so funded through the NRC, and this is a project through my lab at UBC and the NRC, we've committed to getting, and we're making great progress. By the way, it's already a proof of concept as it's been done before. We have a better vector. They're also setting up manufacturing, by the way, I hear also in Edmonton and Montreal, that will serve as manufacturing capabilities for gene therapy for Canada. Because if you can improve the manufacturing capabilities and do it here, so the plan here, and we estimate the cost maybe 30 or 40,000. By the way, you only give a single therapy for the patient's lifetime. So it's ongoing. So so suddenly you're going from, by the way, many gene therapies like for spinal muscular atrophy today cost 3 million Canadian. We would be going down to deciles of thousands. It would be available, single therapy, and we've committed to provide this at cost. So now with the NRC, they're setting up the manufacturing capabilities for the country. This is the first project that they're doing. It's still early, but I think we have a real opportunity. So we would love to be able to bring this to patients. This is a rare disease, but it also talks, there are going to be others. What's next? And so the NRC, together with us and others, are looking at, so what's the next gene therapy? The vector looks very good. There's unique intellectual property that NRC has done very innovatively that allows you also to get protection. And so the commitment's a very novel model for any biotech where you agree that you're going to provide it at cost, overall cost to Canadian patients, but will be commercialized for the rest of the world. So, but Canadian patients will benefit. This work is being funded through taxpayers, and we want to be able to do this. And when you talk to the patients who were treated with this in therapy, it's amazing. It's very inspiring. They tell you their story. This woman who was on national television, on the national, spoke about having 2 children, changed her life with this. And that was only because she participated in the trial. But now also we're able to get, so very importantly, more potency, which means you need less vector. So it's lower dose. and also single injection. We now have data because before it was administered intramuscular. We're hopeful a single injection sub-Q. So procedure, not costing the healthcare system much. And this is just in process. But it's been an amazing collaboration with NRC, and the federal government is supporting this.
Thank you very much. In somewhat of a related question, it says, did you feel Canada had the resources for you to pursue these translational studies? So this refers to the study specifically, or did you have to seek support outside the country?
Yes. So the truth is, drug development is very expensive. And there are things that you can do at a university, but there are things you can't do. And it's very important to not confuse those. Because when you're going to get regulatory approval for a drug, you have to follow a huge number of regulatory compliance issues that the university could never do. There's the manufacturing has to be in a different way, the trials and so on. So the university could never afford the clinical trials. You can't get money to do these clinical trials in Canada because these are costing millions of dollars to get the rigor that's needed for regulatory approval. Now, ideally, we're able to create Canadian companies that can then raise money from the private sector in partnership with the university. But I've had to go outside of Canada because the resources were not available. And the venture capital, Canada has been quite averse to investing in biotech. It's changing now. There have been some amazing stories. I just want to alert people. For example, the Pfizer vaccination, the nanoparticle surrounding that came from UBC Peter Cullis. The first antibody approved for COVID came out of a small company in Vancouver called Abcelera. So Canada's done very well, but you often need to create these private companies that can raise the resources. And in the past, there wasn't the money to support it. That is all changing now.
Thank you for that. Another question, a bit of a more general question. It's a great presentation. There are many gray swans that reflect more than a single gene abnormality. How do we select which swans to follow amidst the commonality and heterogeneity of things like heart failure, diabetes, for example?
Yeah, that's a great question. The problem with gray swans is these are often mild gene and environmental interactions, and sometimes multiple genes. So when you look at some of the diabetes, you really, if you have mild effects of multiple genes and big environmental changes, very hard to think about gene therapy, very hard. You really need to go to those that are extreme outliers. These patients that have, and subjects that have multiple genes, often small, and certainly when you look at genetic contribution to complex diseases from mental illness, schizophrenia, manic depressive illness, there have been like 25 genes, but all of them contribute 3%, 4%. So influencing that gene does not become very helpful. But there are other disorders, for example, disorders of premature aging, progeria, another rare disorder, where now that's looked at, could that prolong healthy life by using that and understanding that mutation leading to treatment, both for these patients, but also for the general population. So gray swans are very useful scientifically. They lead to great manuscripts accepted in the best journals, but they're not great targets for therapy because you're going to leave, when you use a drug, the drug is going to be targeting a single target. That's how you'll screen for it. And so you need that gene to be more than, contributing, at least in these patients. Just think about the statins for a moment. The statins were initially developed in patients with familial hypercholesterolemia, patients with mutations in the LDL receptor. Initially, it was just targeted for patients because they're what you wanted to do. You got upregulation of HMG-CoA reductase to increase synthesis because the cell was short of cholesterol and cells need cholesterol. And so if you inhibited the HMG-CoA reductase with a statin, you got decreased. And initially it was thought to be used in familial hypercholesterolemia. And then it was realized that if you inhibited that gene in all of us, you could lower cholesterol in the general population. So often the initial target will be your selected population, but it could be more widespread. So for example, for the sigma-1 receptor agonist, for Huntington's and potentially ALS, that's great, but the mechanisms involved in cell death in those disorders are associated with Alzheimer's, Parkinson's, retinal degeneration with optic atrophy, and actually some children developmental disorders like Rett's and Fragile X. So once you show a proof of concept and you show that it's safe and tolerable in these selected diseases, it allows you to go to a broader population. But gray swans are great scientifically, but not great drug targets.
Thank you. Thank you for that response. You've had an amazing career, and that was really demonstrated so well in your presentation here today, Dr. Hayden. And so we've got a question for you that says, thank you so much for your inspirational talk, and it really builds on that mentorship. What advice would you have for someone who's in basic science, but is interested in getting involved in translational medicine?
Well, I think, we ideally want to foster physician scientists, and it doesn't mean you need a PhD. Dr. Sikowski at your university has been deeply involved in translation for neurological disorders, and there are others. So I would say, firstly, the basic scientist who wants to be involved needs to understand the disease deeply. And I have found that certainly my early imprinting with patients with Huntington's disease, and where I looked them in the eye and was moved in the recognition that there was a complete vacuum in terms of any therapy, that continues to stimulate and inspire me until we can do something for these patients. And I would say I also like to have my graduate students, with permission of the patients, attend a clinic. Let them see the disease phenotype. Let them understand why this is so desperate. Let them understand why working nights and weekends is purposeful, because they'll have to explain that to their families, because there's always sacrifice. There's sacrifice that each one of us has given in terms of keeping ourselves focused on the goals of our careers. But often families will be hugely supportive once they understand what the purpose is. So I would say helping basic scientists understand why it's important. And if you understand why, the how becomes much easier. And then you also understand what I also see with some basic scientists, they will target a mechanism that either is not particularly relevant or that this is not the big issue for patients. So we have to listen to the voice of the patients. I developed and helped to lead Austedo for Korea. The point of the Huntington patients, actually, to be honest, Korea is not their big problem. And so when we listen to the patients, and by the way, the team came to listen to patients as we do in the clinic here, we recognize that the big worry is their functional decline, not being able to do things at home, not being able to work. If we could give them 6 more months or 8 more months or maybe a year, or the patient with LPL deficiency couldn't get pregnant because pregnancy exacerbated her pancreatitis. Once we understand the focus and the clinical need, and so I would say bringing the basic scientists into the clinic, and then all of a sudden, by the way, the basic scientists got so much to contribute. At the NRC right now, I'm not an expert in vectorology, and so we need to have pooled resources. But if people understand the why, the how becomes possible with expertise around this country. And we can pool that expertise across institutions and across the country, but understanding the why becomes so important. So exposure to the clinical setting and getting permission in our clinics to bring in our graduate students, our postdocs, some of whom are not medically trained, will be hugely inspirational for them. They can go back and tell their families about why they're working so hard and why they're not available for some other things, and that's helpful.
Very wise words. Understand the why, and really it's all about putting the patient at the center of all that we do.
Yeah.
Another question, and this relates to, it speaks to the importance of clinical trials and actually investigating therapies. So the question is, may I ask you about the presented negative outcomes in the clinical trial related to ASO? How much does pharma and the investigators have interest and appetite to go back and fix the problem by drug delivery or other means?
Yes, well, let me say the one message from the failure, and this was really sad and terrible because people had much hope. By the way, there were already signals before this that there were problems. The problems were in the open label for the phase 1/2 study. The neurofilament had gone up. Neurofilament is a sign of neuronal injury, so that was worrying. And the second thing is they had ventricular dilatation. That was worrying because both of those are adverse signs that you wouldn't want. So this related to the methodology, the mode of administration, the failure to reach the target, potential inflammatory response to the ASO. But the actual mechanism of decreasing toxic Huntington still remains a very viable approach if you can get a safe, effective delivery and get throughout the brain, because Huntington's becomes a diffuse disease. So there are approaches. I do know that Roche and Ionis, and for full disclosure, I'm on the board of Ionis. I'm not saying anything public. But Roche, for example, is talking, and they haven't yet pulled the plug on this completely. Maybe there's other approaches. Maybe they could use allele selective, knocking down just mutant, not wild type as well. That's one approach. The other approach is taking a drug, and there are other companies using oral therapy. Now, oral therapy is particularly useful for the CNS development because if it's soluble, it gets to all areas of the brain. So oral therapy is preferred because you know you get target engagement, particularly for a soluble drug, and you know you're able to get the whole brain. So you get to the target. ASO is a problem still because you don't know that you'll get to the target sufficiently. And then, of course, it's a burdensome procedure. When you look at, for example, it's an intrathecal injection. It needs patients to come to hospital. They need to be there at least for half a day. And it takes time and effort. And there's not the infrastructure around the world to treat enough patients. So an oral therapy or different mode of administration would be great. But I think none of this touches on the target itself. Knocking down mutant Huntington represents a useful target if only we could do it safely and effectively. And I'm hoping that, in the end, neurodegenerative disease is going to be like cancer. And we're quite used to that in cancer. Combination therapy, and of course, treating early. There's no point in trying to treat Huntington patients or any neurodegenerative disease in the late phases when they've lost all their neurons and they have no cells to protect. So early is better, and to be honest, in Huntington disease, before symptoms. So we'd love to go from secondary prevention, which is treating patients who are affected, to primary prevention, because we know 100% those patients destined to be ill. And before you have onset of the disease, there are mild symptomatic changes, particularly in motor, but also seen on imaging, that tell you that that patient is close to onset. That would be a great group to treat, because neuroprotective therapy, you want to treat them before the fire has destroyed the house, but where there's just a little small fire so you can diminish that and keep the house intact.
Very, very important approach for sure. We're almost out of time now, so I just want to take the opportunity again to thank you, Dr. Hayden. As many individuals have commented, such an inspiring talk. You're able to incorporate your career journey and your ability to mentor with the great progress that you have made in this field as well. So sincere thank you. Thank you to Dr. Kazam and to Andrew in the Department of Medicine for sponsoring this event. And thank you to all of our attendees as well for attending on this Monday morning. Thank you again, Dr. Hayden, and I'd like to wish everyone a good day.
Thank you all. Thank you for the privilege of doing this. Thank you.
Pleasure. Bye-bye, everyone.